Kyle, I'm a grad student in Dr. Allison O'Dowd's lab at Cal Poly Humboldt, and I'm just going to talk today about the results of my master's thesis, which is attempting to quantify benthic macroinvertebrate food web responses to Klamath Dam removal downstream of Iron Gate Dam. And can I move through here? Yeah. So this study was inspired in large part from the paper done by Sarah Morley and Jeff Duda and others on the Elwha, which found really dramatic declines in invertebrate drift densities during dam removal downstream of the dams. I want to emphasize, this is a really oversimplification of their results. I highly recommend reading their paper. It's easily accessible. But during their spring sample effort, they found really dramatic decreases in energy abundance or energy density in drift samples in the first year of dam removal, which was 2012, and then even more dramatic declines in the second year of dam removal, 2013. You can see here the size of the pie represents the amount of energy available, and then the colors are representing the source, terrestrial or aquatic, of that energy. And by their second year of dam removal in 2013, basically the entirety of their energy availability was coming from terrestrial inputs. So it wasn't aquatic at all, which kind of indicated a collapse of the aquatic food web. As you can see here. So for us, we were really interested in the effects primarily focused on the 60 miles downstream of Iron Gate Dam. That's where we anticipated impacts to be highest. And we collected 2-ish years of before dam removal data and one during dam removal in 2024. Our overall design was we collected data at 4 main stem Klamath sites and then 4 paired tributary sites to use as reference sites, because obviously tributaries do not experience dam removal impacts. And in 2022, we were only able to collect data from these the upper two main stem sites and their respective tributaries. So it was a much smaller data set. For invertebrate samples, we collected 3 types of samples. One was benthic samples, where we used a kicknet, dnet to collect invertebrates on the benthos, and then we collected drift samples, which are pictured here. And these are basically nets that you suspend in the water column. They collect invertebrates naturally drifting down in the water column, and this is generally representative of what's most available to juvenile salmonids. Juvenile salmonids are primarily assumed to be eating in the water column as they're moving through the water. And in the tributaries, our sample design was fairly straightforward. We sampled drift samples at the bottom of a riffle and then moved upstream and collected 3 benthic samples. Each was a composite of 3 kicks. In the main stem, we had to get more creative. Obviously, the main stem's it's a big river. We couldn't pound T-posts in the middle of it, so we had to primarily sample on the margin or in side channels, or sometimes even had to use people as T-posts because the substrate was too loose. We also partnered with the Karuk Fisheries Monitoring Program to collect gastric lavage samples, where we collected 20-ish fish from each site in each year. And this was really cool because we got to actually see in real life what the fish were eating and pair that with what we were seeing in invertebrate samples. So our primary sample season, we were most interested in what was available to fish during peak juvenile Chinook outmigration. I do want to point out that peak juvenile Chinook outmigration often happens pretty early in the spring, and water levels are pretty high and dangerously high, so we had to wait till after peak outmigration. But we did sample in the spring. We collected all our sample types in the spring season. And then I was also interested in seeing if we could detect any differences in responses in the fall, but because of time constraints, I wasn't able to process a ton of samples for this. So I only collected benthic samples, and I only collected these samples in the main stem sites and only in November of 2023 and 2024. That's a much smaller data set. Laboratory methods, the samples all came back to the lab and got processed by an army of undergraduate technicians. For benthic samples, aquatic insects were identified to genus level, but to save time, drift and diet samples were only taken to family level. But we also collected length measurements on each individual in these drift and diet samples to attempt to quantify or estimate biomass using taxonomic-specific weight length regression coefficients. This is the fun part. For univariate metrics like abundance, biomass, I used generalized linear mixed models in a Bayesian framework and just did a very simple before-after control impact-ish study where we tested the interaction of site type and treatment, so main stem versus tributary, and before during dam removal, to see if main stem and tributary data behaved differently during dam removal. For the seasonal effects, because I only had main stem samples and I only had benthic samples, one sample type, I actually tested the interaction of season and treatment on the main stem samples. For multivariate or community composition questions, I used Permanova, permutational multivariate analysis of variance, SIMPR analysis to show which taxa were driving trends, and then NMDS or non-metric multidimensional scaling just to visualize how communities were similar to or different from each other. So what did we see? I know a lot of people aren't familiar with Bayesian approaches, and I wasn't before this either, but I just want to report that I'm not using a p-value here for Bayesian reporting. I'm using what's known as the probability of direction, and this is representative of the proportion of possible effect sizes that are on one side of zero. For simplicity's sake, I'm just going to say that a PD of 0.975 is barely equivalent to a p-value of 0.05. So PDs closer to 1 indicate stronger effect or stronger evidence of an effect, and PDs close to 0.5 are basically non-existent evidence of effect. So I'm going to present all of my metrics here for all of the different samples kind of combined. So for all the samples, the diet, drift, and benthic samples, I looked at abundance and biomass metrics. Biomass, again, I only estimated from drift and diet samples. So for benthic abundance, so the number of individuals in each sample, we basically saw no evidence of change relative to tributaries. That in these plots, the main stem samples are shown in color and the tributaries are black. Oops, that all came together fast. There we go. For drift samples, I did notice an increase in count density, so number of individuals per meter of water that flowed through the net. However, that was not reflected in biomass density. Biomass density basically changed similarly to trends in tributaries on the main stem. For diet count density, so number of individuals per gram of fish that they came out of, I did want to scale these because, you know, larger fish do eat more. We detected increases in both count density and biomass density for diet samples, and these were both significant. I do want to point out that for 2023, our diet samples had a large proportion. I think as many as 50% of our diet samples from 2 of our sites were actually fully empty. We had no stomach contents, and there's a multitude of reasons this could have been the case, but I think the McKinney fire is one option. Unfortunately, we don't have enough data to really assess that statistically. So for now, I just want to keep in mind that a lot of these increases we're seeing in the diet samples are driven a lot by the fact that we had so many empties in one of our before years. Percent Ephemeroptera, Plecoptera, Trichoptera, or percent EPT, for all you bug nerds out there, is a very common metric used in assessing stream health, in part because these orders, mayflies, stoneflies, and caddisflies, are generally accepted or generally seen as more sensitive to disturbance and pollution. And so We looked at percent EPT in all our 3 sample sites and found in our benthic samples absolutely no evidence of change in the main stem relative to tributaries during dam removal. But in drift samples, there was very moderate evidence of change, and this is where Bayesian models kind of have a strength here, as you can kind of indicate that like 92% of posterior draws indicated a directional effect. It's not, if I was using a p-value of 0.05, that's not technically significant, but it is something. And then for our diet samples, we actually noted a very significant decline in percent of Ephemeroptera, Plecoptera, Trichoptera represented in the diet samples from our during dam removal year. And so that's fairly notable. We did ask the percent terrestrial question, mostly because they asked this question on the ELWA, and they did see really strong effects on the ELWA. But for the 2 samples that terrestrials were present, drift samples and diet samples, we did not see much of an interaction in drift samples at all. And we did see an increase in percent terrestrial represented in diet samples. However, some of this I do think goes back to the just number of empty samples that were that we collected in 2023. I also looked at richness metrics, so the number of taxa represented in each sample type. For benthic samples, richness did significantly decline relative to tributaries in our benthic samples, and as did Shannon diversity. So Shannon diversity takes into account number of taxa, but also their evenness across a sample. And so we saw notable declines in both of these metrics for benthic samples. In drift samples, we actually didn't observe any evidence of decline relative to tributaries. And in diet samples, we did again see a very strong increase in number of taxa represented in diet samples. However, this again, I think has a lot to do with the number of empty stomachs we had in 2023. It's just, when you have more bugs in a sample, you have generally a higher chance of getting a lot more taxa. I don't know, this is I looked at community differences using multiple different metrics, but one approach was using non-metric multidimensional scaling, which basically just flattens 3D space onto a 2D plane. And so, as simply put as I can put this, the further apart groups and clusters of points on these plots are, that means that indicates a bigger community difference, right? And so for here in our benthic samples, I see a much stronger difference between 2022 and actually the other 2 years, 2023 and 2024. I've colored these polygons. So the colored polygons represent different years. The different shapes on this actually represent different sites for each main stem site. And then the dashed lines are outlining those clusters of sites. So for the benthic samples here on the left, 2022 is kind of the centroid for that cluster is really far off from 2023 and 2024. And then the sites are actually further apart. Each site has its own unique polygon and cluster. So what that tells me is site-specific differences were bigger than dam removal effects. And then for drift stuff, for the NMDS, you can kind of see that the drift samples all change, kind of each site changes in a different direction. So it kind of expands a little bit during dam removal. And this was reflected in our Permanova models, which showed significant changes in drift and diet main stem samples relative to tributary sample communities, but no significant community shifts in benthic samples. And that goes back to, like interannual differences and site differences in the benthic samples were just a lot stronger. So I ran what's called a SIPR analysis to really assess what taxa might be driving these community shifts and in the drift and diet samples. And what's interesting is across the board in the main stem samples, these taxonomic shifts were driven by the same 3 or 4 taxa. However, in tributaries, any significant changes were not comparable across samples. They basically were fairly random. These 3 taxa 4 taxa really are 1 mayfly family, and then 3 true flies or dipteran families, which are black flies, simuliids, and chironomids are non-biting midges, and blepharosaurids are net-winged midges. And so it's kind of interesting that that was consistent across both sample types. These shifts, though, did happen. They were very site-specific. So if I break it down by site, and for these 4 taxa of interest, you can see the diet samples on the bottom and the drift samples on top here, where drift samples had a huge increase in chironomids at our uppermost site at Iron Gate. We saw a comparable increase in chironomids in the diet. The same goes for blepharosaurids. When they increased at our Klamath at Gottville site, you saw the biggest increase in diet representation in diet samples at the same site at Klamath at Gottville. And the same goes for simuliids. So the increase at Klamath at Gottville was represented in both diet and drift samples. So it's just interesting. This is much more site specific than we were expecting across the board. But there's a lot of site differences between these sites. So that was our spring samples. That was like the bulk of the study. And I wanted to put it on a timeline here relative to dam removal. The bars on this plot, I have sampling windows already laid out here, and then this red bar is representing when reservoir drawdown was mostly happening, and that's really where we anticipated the bulk of the impacts to occur, was from those, the flush of sludge coming down. And so we sampled a month or 2 after the peak elevated turbidity in the river for our spring samples. And then we also took that smaller subset of samples in the fall after coffer dam removal. So we see here the spike in maximum daily turbidity occurs again in September and October during coffer dam work. And we sampled in November, so like a little less than a month after that. So the fall results are a little interesting. Because we only took benthic samples and we only took mainstem samples, I actually, instead of comparing tributary and mainstem samples here, I compared fall samples and their response during dam removal to spring samples and their response during dam removal in the mainstem. And you can see here that while we did not see a decrease in benthic invertebrate abundance in the spring, which is green, we actually saw a really notable decrease in the fall. And so that's really interesting. We saw a response we did not see in the spring, we did actually see in the fall. For the other 3 metrics, percent EPT, taxa richness, and shannon diversity, all 3 of these things seem to decline fairly comparably in the spring and the fall. Although I want to note percent EPT declined more in the fall than in the spring, as did taxa richness. This is represented in this NMDS visualization, where you can see the community clusters for the 2 spring groups, so before dam removal and during dam removal, are much closer together. These are the blue ones up top, and then the fall clusters before dam removal and during dam removal are much further apart. So we see a bigger community shift overall in the fall relative to before dam removal. Again, running a SIMPR analysis, this was primarily driven by a major decline in 1 taxa, our simuliid black flies. These guys generally need high oxygenated water, and I think what's probably likely happening is this decline primarily happened at Iron Gate, and coffer dam removal tanked dissolved oxygen at that site. And because there were a lot lower flows occurring at that time, it probably stayed lower and caused higher impacts for sensitive taxa at our Klamath at Iron Gate site. So just in summary, our top taxa driving changes before and during dam removal were these 3 dipterans and a mayfly that I haven't included. And they all have kind of varying life histories. Chironomids are often recognized as fast dispersers and fairly sediment tolerant, and I think that's why we saw them increase so dramatically at our Iron Gate site, which is the uppermost site right below the dams. Simuliids and blepharosaurids, they're both substrate. They really require substrate for attachment. These net-winged midges on the bottom, all of those little dots on them are sectorial discs, and they really need to glue down to the rock. So if there's no substrate available, like for instance, if sediment had actually stayed in the system for a lot longer, we would have expected to see a decline in these individuals, but that was not the case, I think, because sediment was moving through the system way faster than we expected. So take homes, we did not detect nearly as strong a negative impact on juvenile salmonid prey availability during spring sampling. This really contrasts with the ELWA findings. Obviously, there's a lot of differences between ELWA dam removal and Klamath Dam removal, but we were very surprised by this. Community composition changes in the spring drift and diet samples were primarily driven by increases in these 3 dipteran taxa. And dam removal effects may have been more dramatic in the fall than the spring. Unfortunately, we don't have samples from the fall for other sample types, and nor can we compare changes in the tributaries to mainstem sites. So it's a little bit weaker, but I think future monitoring may shed some light on that. I want to thank the Crew Track Fisheries Program for collecting all our diet samples, my advisors and committee members at Cal Poly Humboldt, amazing lab technicians, and then we also partnered with the UC Davis Lusardi lab for a different part of this study. So, and are there any questions?
Now, let's not have everyone ask questions all at once.
That's the last part of the day. So
yeah, that might be part of it. Laurel, and then Chauncey.
Yeah, awesome talk. Lots of really cool information there. I'm curious if you had ideas about the specific mechanisms at play in the ELWA, and because dam removal has so many different things that's happening, and it's different, like you mentioned briefly, but what was the specific mechanism of the ELWA that caused the huge decrease? And is that mechanism something that we also had on the Klamath?
Yeah, no, that's a really, really great question. I think, so I want to point out that neither my study nor Sarah Morley's study had sufficient data to really, or well, I should say my study did not have sufficient data to to really model habitat shifts. So this is all completely conjecture. But the Morley paper did propose a couple of mechanisms. I think one of the big ones is they had multiple years of sediment impacts, right? They were removing dams over a series of years, and I believe, correct me if I'm wrong, I'm sure people on this talk know better than I do, but they removed the upper dam first and put a lot of sediment into the lower reservoir, and then flushed that sediment down the second year, and it really impacted, there were basically just years of compounding sediment impacts. Additionally, the ELWA, the sediment in the reservoirs was dominated by larger substrate. So we had a lot of fines that stayed elevated in the water column and moved out really fast. And so, there's a lot of differences, obviously, between these 2 rivers. And the dam removal projects. But I do think that it's really interesting that the timing of the dam removals on the Klamath, in my opinion, really did maximize river health, with timing it with what would naturally be river, big pulses of water coming through and pulses of sediment coming through. So personally, I would attribute it to just finer sediment, less more mobilized sediment, and shorter duration of sedimentation impacts in the actual water. Obviously, if any of you have been out there, there's a lot of sediment that's piled up on shore for the river, and it's that's going to take a minute to flush out. But really, when we sampled, we were noticing a lot less sediment in the river, in the substrate, than we were expecting.
That's super cool. It's a really neat indicator of just how robust the river is at being able to handle these disturbances and recover really quickly. So super cool to see that. Thank you.
Chauncey?
Oh, want me to call you? Sorry. Chauncey, do you want to?
Yeah, just that was really good, really interesting. I didn't catch much about the McKinney fire, and I couldn't quite remember what your sampling timing was relative to McKinney, and so can you talk about how that might have affected any of your findings?
Yeah, that's a great question. So unfortunately, because we only have 2 data points from 2022 in the main stem, One of those was above McKinney Fire footprint. So we have our Iron Gate site, and then we have our Klamath at Gottville site. The only one that experienced any impacts from from the McKinney Fire was that Klamath at Gottville site, which means I have 3 samples to work with. I couldn't run any analyses, basically, testing for effects of McKinney Fire. I do think that it's very possible that our 2023 diet samples had low proportions of invertebrates in the stomachs, maybe in part because of the sediment effects that were happening at those sites. I don't know how the McKinney Fire sediments were there was a lot of sand, a lot of gravel, a lot of stuff stuck in place a lot longer, and so I think substrate and bug habitat might have actually been more affected by McKinney Fire than dam removal. That's all conjecture. I can't actually prove it. Yeah.
Before we jump to Beat King, I want to make a quick just point. If anyone's new to KBMP, we want to know who is attending. I'm assuming that's Ben King, but if you're new to KBMP, please put your name and email in the chat so we can track who attends and we can stay in touch with you. You can do direct chat with me if you want to not share it publicly. That's fine. But just want to put that out there because it's really important for us to be able to know who attends. For example, I see Lacey and Lacey Green. Sorry to point that out, but we don't have any Lacey's in our database of names, so apologize for that, but just want to make that point, and it's important. I'm assuming that's Ben. Please go ahead.
Yeah, sorry, Randy. I didn't get a chance to change my Zoom name. I don't use Zoom particularly often.
Yeah.
But that was awesome, Rosa. I'm glad to finally get to hear some of the synthesis of all of your hard work. I was wondering if you tested for kind of specifically a result of proximity to Iron Gate. I think it comes out a little bit in some of the previous questions about how the Klamath itself processes sediment compared to the Elwha just on a distance level. But I was curious if you tested for that specifically.
That's a great question. I really tried. I spent months and months trying. I just didn't have quite enough data to get the models to run cleanly. But I did see, the reason I don't report it is because basically 3 metrics popped as significant when they really didn't, there wasn't sufficient data for that. But taxa richness and diversity were very dramatically tied, or the impacts to those metrics were very dramatically tied to dam removal. Distance from dam. So yes, the biggest taxa richness and diversity impacts occurred closest to the dam, and that definitely diminished as we went downstream. One of the issues with our sampling was our lowest sample site on the main stem. It was just a crappy site, to be honest. Like it was the one where we had to stand with our drift nets and hold them, and it basically the quality of the data, in my opinion, and the quality of the samples actually decreased as we went downstream too. So it's all convoluted, and but yes, it's a really good question. I really did try to get to statistically test for those, but I can tell you anecdotally that distance from dam did really, really affect the number of taxa represented in the samples.
Sweet, thanks.
Yeah.
Is it too early to refer to you as Dr. Rosa Cox yet?
Yes, 1, because I am not getting a PhD, and 2, because I am not done.
I'm sorry about that. I knew you were defending your
Thank you.
just this week, so.
No, I actually had to push it back. I'm currently working a full-time job, and it's amazing how distracting that is.
Okay. Great. Well, if there are no other questions, I want to thank Rosa, and I want to thank all the other speakers who've gone through today. It takes a lot of time to prepare a presentation, so when you volunteer to give a talk and share your amazing information with us, it just helps to make sure that we all know a little bit more than we did this morning, and how we take that information and incorporate it into our work and how we evolve our own studies and approaches to restoration or monitoring, I think goes a long way to where we are now. It's been an amazing journey here on the Klamath in the last few decades. So thanks to everyone there. And I want to thank everyone who just has stuck with us for the whole day. It's been a lot of great talks, and you guys have been great listeners. So thank you there. If anyone needs help getting in touch with a speaker or other people who's attending today, just go ahead and reach out to me. We can help connect you.